Hello, I am JD and you are listening to The Ongoing. Our planet is covered in water. Oceans, seas, rivers, and lakes. Water is everywhere. But most of it, about 97%, is salt water. And salt water we cannot drink. For most of human history, that was simply a limit we lived with. This episode is the story of reverse osmosis, how we learned to remove the salt from ocean water and make it drinkable. The technology that made it possible is called reverse osmosis. And the idea behind it came from nature. From a principle that every living cell in your body uses right now. So let's begin. In 1748, a French priest named Jean-Antoine Nollet was doing experiments in Paris. He was one of the most celebrated science demonstrators of his time, known for making complex ideas visible and dramatic. In one experiment, he took a pig's bladder, filled it with alcohol, sealed it carefully, and submerged it in a container of water. He expected nothing unusual. The bladder began to swell. Water from outside was moving through the wall of the bladder into the alcohol. The alcohol could not escape. Only water could get in. The bladder kept swelling, and in some experiments, it eventually burst. Nollet was puzzled. He had not added any pressure. He had not stirred anything, and yet water had moved on its own through a solid wall. He had no name for what he was seeing, but he had found something important. The wall of the bladder was behaving like an extremely fine filter. So fine you cannot see it. It had openings, invisible to the naked eye, small enough for water molecules to pass through, but not for large molecules like alcohol. Scientists would call this kind of a barrier a membrane. And what Nollet observed was that water didn't just passively sit on one side of the membrane, it moved on its own toward the alcohol. Because the alcohol side was more concentrated. There was more dissolved in it than in the plain water outside. And water naturally moves towards the more concentrated side, as if trying to balance things out. Over the next century, scientists studied this across nature. In plants, in animal cells, in the human body. And they gave it a name: osmosis. Water moving through a membrane from a less concentrated side to the more concentrated side, naturally without any force. What Nollet had stumbled upon in his Paris laboratory turned out to be one of the most fundamental forces in biology. Every living thing on earth depends on it. Your body relies on osmosis every minute of every day. Think about your kidneys. Every 30 minutes, your kidneys filter your entire blood supply. Here is how it works. Blood flows into the kidneys under pressure. Inside, there are millions of tiny filtering units, each one built around a membrane, the same kind of selective barrier that Nolet had observed in his bladder experiment. The membrane lets water and small waste particles pass through. Larger molecules, proteins, blood cells are too big. They are blocked. The waste that passes through becomes urine. The clean blood continues circulating. All of this happens automatically. Nature had been running this for billions of years. And once scientists understood it, they began to wonder, if a membrane can separate waste from blood, can it separate salt from water? That question had a name: desalination. Now, in osmosis, water moves naturally toward the salty side. That is what nature does. But for desalination, we need the exact opposite. We need to push water away from the salt through a membrane in reverse. And that is reverse osmosis. And it requires a lot of pressure to force it through. Reverse osmosis is the same process that filters water in your undersink home filtration systems. Not just to remove salt, but to remove lead, chlorine, pesticides, and other dissolved contaminants. To build desalination plants, the first challenge was the membrane itself. A membrane is a thin layer that separates two substances. What makes it remarkable is that it is selective. It does not let everything through equally. Some molecules are small enough to pass, others are too large and are stopped. Think of it like a filter with openings at a scale we cannot see. The membranes nature uses are living. They cannot be scaled up. Scientists needed to build something artificial, a synthetic membrane, one that could replicate what nature does, but that could be made in a laboratory, and eventually at a scale that could supply millions of people. Nobody had ever built anything like that before. In the late 1950s, two researchers at UCLA, Sidney Loeb and Srinivasa Saurirajan, made a breakthrough. They developed the first practical synthetic membrane made from a material called cellulose acetate, with pore sizes small enough to block salt while allowing water to pass through. And it worked. But working in laboratory and working in the real world are two different things. In 1965, reverse osmosis was tested for the first time outside a laboratory with the help of UCLA researchers. In a small town in California called Coalinga. The groundwater there was so loaded with minerals, it was undrinkable. The town had been importing its drinking water by train. Reverse osmosis system was installed. Water was pushed through the membrane under pressure, and on the other side came clean water. For the first time, the town had another option. That movement proved reverse osmosis worked, not just in a laboratory, but in the real world. To scale up desalination plants, thousands of membrane units needs to be manufactured. Identically, consistently, every single time. The spaces inside the membrane have to be precisely the right size. Large enough to let water molecules through, small enough to block salt. Too large and salt gets through. Too small and almost no water flows. That margin is a fraction of a nanometer. There is also the question of porosity. How much of the membrane surface is actually open for water to flow through? Too little porosity and you need enormous pressure just to get water moving. Too much and salt starts to get through. That balance had to be engineered, tested and reproduced at scale every time. The original membrane was made from cellulose acetate. Over the years, chemists and engineers developed better materials, including a layered material, a thin polyimide film bonded onto a porous support. Thinner, more durable, more effective at removing salt, and manufacturable at scale. And running alongside all of this was the challenge of energy. Pushing water through a membrane under high pressure requires an enormous amount of electricity. Making that process efficient enough to be affordable was its own long engineering problem. The technology, though, kept improving and the world began to depend on it. Saudi Arabia is one of the driest countries on earth. Almost no rivers, almost no natural fresh water. Today, more than half of all the water Saudi Arabians drink comes from the sea. Without reverse osmosis, Saudi Arabia could not sustain its population. Israel faced a similar crisis. By the early 2000s, its main freshwater reservoir was dropping year after year. The government built 5 large desalination plants in a decade. Today, more than 80% of Israel's household drinking water comes from the sea. A country that was running out of water built its way out of the problem. What started as a French priest watching a pig's bladder swell in water became one of the most consequential water technologies in human history. But the distance between that observation and the desalination plant supplying a city was engineering. The science told you it was possible. The engineering was the part that was actually made it possible. That's it for today. Thanks for listening. I am JD and this is The Ongoing.